All right, so this is part 4 of chapter 1. We're moving on to macromolecules. So, in comparison, when we talk about macromolecules, these are pretty large. We have 4 main types. We have carbs, or carbohydrates, lipids, proteins, and nucleic acids. This table, you can use this as a reference. I have a little note up here in the corner to use this table as a reference. It's got a lot of really good information, and there could be questions taken from the examples right here, this column, or notes about examples. I could ask questions on the test from either of these columns, and I don't expect you to necessarily memorize this, and that would take a long time to do probably, but if you have it handy and use it as a reference, then that's helpful when we're talking about being assessed on it. So I just when we're talking about these macromolecules, think of them and how they participate in making up the microorganisms. Okay, so monosaccharides, or not necessarily just how they're made up, but also how they contribute to these microorganisms, if that makes any sense. So monosaccharides, sugars that are involved in metabolic reactions. They're the building blocks of disaccharides and polysaccharides. Disaccharides, the next thing down, they're composed of 2 glucoses, an important breakdown product of starch. Polysaccharides, they make up the cell wall and also are used for food storage. Lipids are the major component of cell membranes. Phospholipids, also you should know that they are the major component of the cell membranes. Triglycerides, fatty acids, the whole 9 yards there. The waxes, when we talk about waxes later on, they make up part of the cell wall in mycobacteria, which is a type of bacteria. Steroids are in membranes of eukaryotes and some bacteria. Proteins serve as structural components and perform metabolic reactions, and we'll learn later on that proteins, there are different examples of proteins include antibodies. Antibodies are proteins. Enzymes are proteins. Enzymes are needed in metabolic reactions. So hopefully you'll be able to remember that. And there's some other examples here listed too as well. And then we have nucleic acids, specifically DNA and RNA. So DNA mediate inheritance. Basically, they have the secret code to pretty much make up any specific cell or any specific organism, and RNA facilitate that code. They, kind of like a doctor and a nurse, right? An RN and a doctor. D for doctor, RN for RN. The doctor has has information that he passes on to the nurse, and then the nurse facilitates that. The doctor says, I want you to perform, send the patient for a CT scan. Then the nurse, basically follows those orders. So just, again, use this as a reference. I might ask you what makes up a monosaccharide. Glucose makes up monosaccharides. Fructose. What is a disaccharide? Maltose, lactose, sucrose, polysaccharides, starch, cellulose, and glycogen. Okay, so just note these 2 columns in reference to these macromolecules here. So carbohydrates are a combination of carbon and water. Now, I can tell you, I'm not going to ask you for the chemical symbol or the formula. It just hurts my head to get into the chemistry of it. So, a lot of this you can kind of skip over. Some of this I left in as a reference, and there may be some test questions related to these. So that's part of why I left these in. So I'm not going to necessarily read through every single one of these slides to bore you, because you can reference it yourself. Polysaccharides, these contribute to the structural support and protection of the cells. They serve as nutrient and energy stores. Cellulose is the cell wall of plants and some microscopic algae. Agar, or Agar, some people call it agar, I call it auger, is an important component of culture media. So when we're talking about being able to grow bacteria, we have to have some sort of solid surface to do that on, and we use auger as part of that. Sometimes we add all sorts of good stuff into it to make it more appealing to certain bacteria, or we put things in it that keeps some bacteria from growing, but that's a whole nother story we'll get into later. Chitin is the part of the cell wall of fungi. Peptidoglycan, which is my favorite word in microbiology, is a component of of bacterial cell walls. And we're going to talk a lot about peptidoglycan when we get into gram staining and such. Lipopolysaccharides are a component of the gram-negative cell wall, and these guys can cause all sorts of havoc in a person if they are infected by a particular gram-negative cell bacteria that contains the lipopolysaccharide. The glycocalyx, should have heard about glycocalyx in AMP1. It is a sugary kind of polysaccharide layer. In bacteria, it's a protective outer layer, and it plays an important role in the attachment of cells to other cells or surfaces. So it's like the glue on the outside of a cell. And one thing that we'll learn is that in order for, there's a lot of barriers in a person to prevent them from getting infected with a particular microbe. But things like the glycocalyx will ensure that microbe of taking a good hold on the cells of the person or the animal or whatever it might be, to be able to infect them. Lipids or triglycerides, important storage lipid, composed of a single molecule glycerol bound to 3 fatty acids. Those fatty acids can be saturated or unsaturated. Lipids are stored in long-term concentrated form as droplets or globules. And it's important to note that while we do get a majority of our energy, a lot of energy, we'll say, I don't want to say majority, but a lot of our energy from carbohydrates, as well as other living organisms get a lot of their energy source from carbohydrates, lipids actually pack more in their punch. So they yield twice as much energy per gram as other storage molecules. So specifically carbohydrates. So lipids, are worth more essentially when we're talking about energy than carbohydrates. Phospholipids and membranes. So again, a lot of this should be review. We have hydrophilic, hydrophobic. Hydrophilic is water-loving. Hydrophobic is water-fearing. When we're talking about the membrane lipids, the hydrophilic part is the head, and the little feet, the little tails, are dangling on the outside of that cell membrane. And so that membrane, the center of the membrane, has those 2 little heads, and the inner and outer part of that membrane has the water-fearing tails. Sorry, I think I have that backwards. Wait a minute. Yes, I have that backwards. The water-loving head is on the outside. The water-fearing tail is on the inside. So you have the tails on the inside of that membrane, and then on either side of it is those little phospholipid heads. So, and this kind of explains it here. When exposed to an aqueous solution, the charged heads are attracted to the water. Nonpolar tails are repelled. So they just naturally assume a single or double layer. A bilayer is what we're familiar with coming from anatomy. That double layer, bilayer, is how they naturally form. It's how they orient themselves. This behavior allows them to be the main constituent of all cell membranes. And there's a picture of the water-loving head and the water-fearing tails. Phospholipid. And when you put 2 of them together, you can see the heads here and the heads here, and then those tails are in the center. Kind of looks like one of those little, like a Kit Kat thing going on there. All right, steroids and waxes. Steroids are complex ringed compounds found in cell membranes, and also they act as animal hormones. Cholesterol is a type of steroid, and that helps to reinforce the cell membrane. That's a test question, I'm pretty sure. Cholesterol is a steroid, and it helps to reinforce the cell membrane, and mainly in animal cells and also cell wall deficient bacteria. And we'll talk more about those type of bacteria later. Waxes is basically what makes waterproofing possible in, say, fur and feathers and fruits. But it's found, certain waxes are found in the cell wall of bacteria that cause TB, tuberculosis, and leprosy. And these waxes that are found in the cell walls of those particular bacteria help to contribute to their virulence or their disease-causing potential. So they're more likely to cause disease because they have this wax helper that allows them to do that. All right, proteins. Proteins are the predominant organic molecule in cells. Proteins, generally speaking, depending on how big we're talking, composed of usually 20 different amino acids. A short chain of amino acids is referred to as a peptide, polypeptide, 20 or more, and then a protein contains a minimum of 50 amino acids. Protein structure, again, this is some review from anatomy. We have the primary, secondary, and then the next part will be the, the tertiary and the quaternary. So primary, simple, I think of it as like if you're crocheting and you just do one long string of crocheting, that is a simple, it really doesn't do much. It's not a very important protein, right? If I just crochet one long string of yarn, then it's not very useful. A secondary structure would be one that maybe I've linked a whole bunch of lines of crocheting strings together, and now I have a really light blanket, right? A very small throw. Okay, that would be a secondary structure. It's a little bit more complex than the primary structure, but it's still very basic. And then the tertiary structure basically would be like, okay, let's put some lining on that blanket, right? Make it more useful. So there's more complicated things related to that blanket, right? It took a little bit more to make that blanket. That's the tertiary structure of proteins. And then when we get to the quaternary structure, that's basically that we'll put heating elements in the blanket, right? It's super complex. You got to plug it into the wall for it to work. It's got all these different colors on it. It's got some fringe on it. Super complicated blanket. Okay, that is the quaternary protein structure. And basically, the same way the blankets have simple tasks and then more complex tasks, that's the same thing with the protein structures. The more simple the structure is, as in primary and secondary, the less important the task is, right? The less complex of task it has. The quaternary structure of a protein, super important, very complicated tasks like enzymes and antibodies. Okay, so when we go from primary, secondary to tertiary to quaternary, it's building upon itself as far as how complex of a job it's going to have in whatever organism it finds itself. More protein structure and diversity. So each protein develops a unique shape, and this is kind of more what I just said. Develops a unique shape, and its surface displays distinct pattern of pockets and bulges. Proteins can only interact with molecules that fit its particular surface features, like a lock and key. So enzymes are catalysts for all chemical reactions, and antibodies, of course, we know about those. They help us fight off stuff that we maybe have been exposed to in the past. So I will pick up on the last part of, hopefully the last part of this, this chapter in part 5.